So backtrack. Hematopoiesis basically is a process of making cells. It's done in microenvironments that we just talked about. Basically, we'll talk about certain cytokines that are known as growth factors, and that's basically the whole process that we're going to talk about with the hematopoietic stem cells, and that's the cell that's going to start all this as we go. Okay, so differentiation basically is the next thing that we need to talk about. So the appearance of different properties in cells which are initially equivalent. Okay, so what that means is when we talk about differentiation, the HSC appears to be like a tiny lymphocyte. If you were to find a stem cell on a bone marrow slide, you would of course have to do flow cytometry to determine that that's what it is. But it would look like a lymphocyte. It would look like a real tiny lymphocyte. Okay? So differentiation is basically all the process that gets it to an RBC, a neutrophil, EO, baso, lymph, T and B, and then there's also a natural killer, then platelets. Okay, and we know that all of these have a different look to them, and then monos in there also, monocytes. All of these look different, but yet this guy looks exactly the same all the time. So differentiation is when we go through the details that go from here to here. Okay? And that's what this whole chapter is about. What it takes to get it from this form to this form. Okay? And we're going to talk about each step as we go. Commitment is when two cells derived from the same precursor take a separate route. Okay? So commitment, all right? So what's going to happen, and I'm just going to go ahead and throw this in here. There's another cell right here, the next step. It's called an MLT, okay? Multi-potential progenitor, multi-potential precursor, whichever way you want to say it. I don't really care, okay? Because it's the same thing, all right? This cell, all right, from here to here, there is no differentiation, okay? Matter of fact, this cell, and we're going to talk about this in just a minute, is never changes. It never differentiates, ever, okay? It stays the same all the time, all right? But through the process of mitosis, it actually creates two of those MPPs, okay? At this point is when growth factors and cytokines start to differentiate them, okay? So what these guys can become is they can go into what we call CMPs or CLPs, okay? CMP, we're going to talk about in just a minute, so you don't really have to know it at this point. But my point is, when commitment, this is commitment, okay? So now that this cell has committed to one of these other ones, okay, this one is going to be those guys, one of those guys from then on out, based off of the cytokines that it gets influenced by. Does that make sense? Whereas this guy is going to become one of those. So CMP stands for common myeloid progenitor, okay, or precursor. I don't really care, whichever term you want to use. I think your book uses progenitor, okay? Common myeloid progenitor is what it stands for, okay? And then CLP stands for common lymphoid progenitor. That's it. Okay? That's what they are. But right here is where we deal with commitment. Okay? Alright? Now once they commit to common myeloid or common lymphoid, there's no turning back. Does that make sense? Okay? So once they commit, it's in this first step with the MPP, okay? Then once it commits to one line or the other, there's no going back, okay? All right? Now, this guy has committed to the myeloid series. Does that mean it's going to become a neutrophil? No. It can become any of these other things that's in that myeloid series. Does that make sense? Okay. Same way with this guy. He's committed to the lymphoid series, but based off of the body's needs, it'll then be influenced further to become BT or natural killer cell. Okay? But it is going to be in that lymphoid group. Does that make sense, guys? Okay.
What did MPP mean?
Multi-potential progenitor or precursor. Yeah, there's a big chart right there in your key. And there's a key in your book too, Kim, if you've got your textbook. Alright. Questions about differentiation and commitment. The two differences in these guys. We good?
Yes.
Right. All right. Now, maturation, okay? Maturation is the entire process of commitment to differentiation to mature, recognizable characteristics, okay? So, essentially, maturation starts from here to whichever one this guy decides to go to. Okay? Alright? So maturation, like we talked about yesterday in lab or in class, is rubriblast through erythrocyte. All of those stages are part of maturation. Okay? And as we go through hematology, you're going to hear me talk about, well, it's going through this particular maturation period, and those kind of things, okay? So it's very important that you know we're talking about basically from that blast stage down to whatever mature cell we're looking at, okay? All right.
Where would I find a rubriblast?
You would find a rubriblast right here. Remember, but it's going to be, remember those names I told you, it's got different names? So it's going to be a pronormoblast. Okay. Remember I told you there's three names, and I always say the old name because I'm old, but you need to learn that blue name. So it's called a pronormoblast. And then the next one, what is it? Basophilic normoblast?
Yeah.
And then the next one is polychromatic, and then orthochromic, and then
Polychromatic
erythrocyte. It changes it from normoblast to erythrocyte. So that means it's lost its nucleus at that point. So it's a reticulocyte, right? And then the last one, of course, is still an erythrocyte. All right? So I'm trying to learn them.
You said maturation?
Yeah, that's what maturation is. Would be from that pronormoblast to the erythrocyte. Okay? All right, so hematopoiesis, two primary characteristics. All right? First thing is it produces a variety of distinct blood cell types per use. Okay, so that's what we talked about. So HSC is going to make us all of these cells by the time we get to maturation. Then, the second thing about hematopoiesis is the relatively brief lifespan of the individual cells. Okay, so we haven't really talked about this, but red blood cells are about 120 days. Okay, so from commitment here to this CMP, a red blood cell, once it goes CMP, basically it goes through a couple more maturations. We're going to talk about those. But CMP to the pronormoblast or the rugoblast form, it's about 120 days lifespan. Okay? After 120 days, somewhere about, day 75, it'll probably get kicked out of the bone marrow and stay in circulation. All right? So, does that mean every 120 days I lose my red cells? No. Normal, you lose about 1% a day. Okay? 1% of your red cells a day is what you lose. Okay? Which is not anything to hurt us. Okay? Especially us guys. Okay? So usually don't have any problems with that. Alright? Neutrophils, a few hours. I don't know like an exact number. But a few hours. EO and basos, a few days probably, because they kind of last a little longer just because of their job. Now T cells though, they can live a lifetime, depending on which ones and what they do. So there's a little bit of variation in your lymphocytes. Okay. And then platelets are just a few days, actually. Like 10 to 15 days, maybe. And then monocytes, they live a long time. I don't know, 15 to 20 days or so. But remember we said they don't stay in the peripheral blood. So that's one of the reasons monocytes live longer, is because they don't stay circulating our peripheral blood. Same way with neutrophils. Neutrophils don't stay in our peripheral blood very long. They migrate, okay? So, y'all are all young. I'm 46, and I don't understand why I still get pimples. Trying to figure that little concept out. I figured my hormones are kind of balanced out at this point. But you guys are all younger, so your hormones are still adjusting and different things. But when you get a pimple, and you get that white center, that's neutrophils. If you pop that thing and put it under the microscope and stained it, it would be white blood cells. It's neutrophils. It would be SEGs. That's what they do. And that's not your blood, right? So it's tissue. And so neutrophils don't hang out in circulation that long, just a few hours. Once they egress from the bone marrow, they don't stay for just a few hours, and then they go into the tissue also, okay, to help with the macrophages. All right, any questions about that so far, guys? Okay. All right, so circulating cells are, all right, now when we talk about circulating, all right, anytime you see the word circulating, we're talking about what? Peripheral blood. That's exactly right. Okay? We don't circulate in the bone marrow. How are you? Where you live?
Birmingham.
Where at Birmingham? I do. Dang, girl. You come a long way. So, did you just not even try yesterday?
I didn't feel like driving Uber every day.
No. Well, what about Terrence? Well, Terrence, where the actual MLT program is. Which I'm glad you're here. I mean, don't get me wrong. I'm just saying that's a long drive. So, 59's in the butt.
Yeah.
You got to go through malfunction junction every day.
Yep.
That's your heart. Yep.
What is that?
So, huh?
That's bad.
It is. All right, so circulating cells are always mature, okay? So, with the exception of one, what did we say? What's one cell that we said we can see 0 to 5 of that we don't get concerned? A what? Nope. 0 to 5. And I said this cell's immature. He doesn't really do the job. What? Nope. No, EOs are usually mature too. I said this is like kicking the 17-year-old out and making him go to work. He can do the job, but he ain't real good at it. You see an increase in this? I told you guys, you always have to be careful about this one. Call it the more mature cell because the doctors start freaking out and thinking they have pneumonia. Band. Okay? So you can see band cells. Bands are technically immature. Okay? They're like the 17-year-old. Okay? They're mature enough to be out of the bone marrow, but they're just not real good, and you shouldn't see that many of them out of the bone marrow. Okay? So the majority of the cells, though, should always be mature cells. They shouldn't have to grow anymore once they get in circulation. Okay? All right, so logical question. If I see immature cells, is there a problem? There is, always, no matter what. Okay? Now, it may not be a detrimental problem, but it is a problem. It is definitely not a normal thing to see immature cells, whether it's in RBCs, immature neutrophils, you know, those kind of things. We just don't see those as we go. Okay? All right? Incapable mitosis. All right, so we're going to talk about mitosis. Y'all remember mitosis? We all taught PMAT. Does anybody remember mitosis? What is mitosis? Huh? Just give me a general definition of mitosis. Very simple definition. Cell replication. That's it. Cell division. That's the good definition of mitosis. So what does PMAT stand for? Go ahead and say it. I want to see how you say it. Huh?
Oh, yeah.
Huh? I wanted to see how you would say it, though. How do you say it?
What?
Do you call it telophase? Because I had a professor at JSU when I was in biology. He called it telophase. And I asked him, I said, well, do you call it the telophone? And he said, well, no. And I said, it's the same damn thing. Telophase, not telophase. Right? It's telophase. Prophase, metaphase, anaphase, telophase. These are mitosis. Okay? And the whole purpose of mitosis is to create what? What would be the term? Y'all remember the term? Say it loud. Daughter cell, right? And a daughter cell should be what? Should be different, identical.
Oh,
Identical, always, right? We don't ever want any kind of mutation to occur from this cell that divides into two cells, right? We want those daughter cells to always be the same, all right? So, up to a certain point, every cell that we're going to talk about goes through mitosis, okay? And not only does it go through mitosis one time, but it goes through mitosis hundreds of times, okay? So, a lot of times on the board, I would say, okay, we got a lymphoblast, a lymphocyte, okay? And you're thinking, oh, that's one cell, producing one cell. No, it's producing 1 million cells by the time mitosis stops. Does that make sense? Okay? As it reaches maturation. All right? So, Need to know prophase. Is there another phase of mitosis? Huh? Say it louder. Meiosis. No. Meiosis is actually totally different. Meiosis and mitosis are both cell division, but in meiosis, okay, it's mitosis twice. Does that make sense? And at the end, you get different cells. Okay? And so meiosis makes meat. You've never learned that in biology. Meiosis makes meat. So eggs, sperm is what meiosis is, the end result of meiosis. You don't want all your sperm being the same and all your eggs being the same, right? So we have this thing called crossing over and different genetic differences in that, okay? But when we talk about, look, I guess I should have said this wrong. I said it wrong, so just overlook me, I'm stupid. I should have said, is there another part of mitosis? Is there another part of cell division that I don't have on the board? How about IPMAT? You ever heard that? IPMAT, what does the I stand for? You remember?
Interface?
Interphase. You go, girl. Interphase. Alright, so, logical question. Where do most cells stay? What phase do most cells stay in? Interphase. Interphase is known as their quiescence stage. Okay? I'm going to put that word up there in just a minute. But quiescence. Okay? Quiescence just means they're resting, is what it is. They're not doing anything. They're just doing their job, kind of hanging out. Alright? Then, once the cell reaches its certain point in interphase, and your body says, okay, I need another cell, then the cell begins to prepare for mitosis. Okay? So if you remember from biology, interphase, or the cell cycle, is in a circle. And you've got, there's a G1, a G2, there's an S, there's a P, and then you have PMAT. And then you have cytokinesis. And then it enters back into G1. Now remember all that from biology? I know it may have been a long time ago. Okay? So like from G1 to the P phase, okay, the cell gets ready to do cell division. So it gets larger, replicates its DNA, replicates its organelles, all these other stuff. So you had a cell that was this size. Normally, when you see the cell get like this, it's in interphase. It's going through a division cycle in interphase. Does that make sense? And then eventually, it'll go through PMAT, and then you'll get those two daughter cells that are the same as the mother cell from the original cell. Okay? This is all review. All right. So limited lifespan or terminally differentiated, so they must be replaced with less differentiated, mitotically active precursor cells. All right? Hematopoietic precursor cells, these are always located primarily in the bone marrow in adults, and they consist of a hierarchy of cells, and that's what we were talking about earlier, the ruboblast, the prorubicite, so forth. Then we'll talk about the myeloblast, the promyelocyte, myelocyte, metamyelocyte, band, seg, okay? Then it's a a lymphoblast, prolymphocyte, lymphocyte. Monoblast, promonocyte, monocyte. And megakaryocyte to platelet. It's actually has a megakaryoblast, pro-megakaryocyte, and then megakaryocyte, and then the platelets, of course, are breaks off of cytoplasm. And we'll talk about that in just a little bit. Okay, so precursor cells, enormous proliferation potential. Okay, daily production. Okay, so this is times 10 to the 11th. Okay, so trillion, got 100 trillion, million, billion, trillion. Yeah, 100 trillion. Okay. So 200 trillion RBCs a day is how much our body makes. Okay. White blood cells about 100 trillion, and same thing for your platelets. Okay. It's a big deal. That's a lot of cells. Okay. That the body has to make. So, of course, can increase production based off of your body's need because of the efficiency that our body has. Okay, so I'm going to take a poll. We just said that RBCs, it takes 20 or 200 trillion a day. White blood cells 100 trillion a day. Platelets 100 trillion a day. Okay? So, how many HSCs do we have? $1 trillion. Who says 1 trillion? I'm asking. Think about it. We've got to make this many cells a day, and they all have to come from one of these guys. So how many do I have? 400 trillion. Anybody say more than 400 trillion? You think it's less? All right. How less? Tell me. What you think?
I think
A trillion would be good? All right. Who says a trillion? Who says higher than a trillion? All right. Who says less than a trillion? Huh? You want to know how many?
How?
20,000. And those 20,000 will last just about your entire life. The average adult has about 20,000 of these. So how can that be? Well, let's talk about it. Okay. Stem cells. All right. Undifferentiated cells, hematopoietic stem cells, what these call them, they give rise to all bone marrow cells by the process of proliferation and differentiation. All right. So, what happens is these guys never commit. Remember a while ago we talked about commitment? They never commit. They're the deadbeat dad. They never commit. You know what I mean? Never get married. Never commit to anything. Okay? So what these guys do is they divide unchanged and become that MPP, multi-potential progenitor. Okay? Now listen to me. This is very, very important. Okay? Once this guy divides, it stops for a little while. Okay? Because he has done cell division to get this guy to start doing this. And make more MPPs. Okay? And then this guy makes more MPPs. And then this guy, and this guy, and this guy, and this guy, and this guy, until eventually, guess what we have? We have 100 trillion potential cells there. Okay? Does that make sense? So, our body efficiently says, why should I make 400 trillion cells when I can keep 20,000 and keep them as my pool? And they can always do the work. Then now, nutritionally, energy, all of the things that a cell needs to live, I only have to put it in 20,000 instead of 400 trillion. Does that make sense? So, genetically, this cell is designed to never be influenced, really, you spell influence. Is that right? F-U, influence. C? Is that what you said? E-N-C-E-D? Okay, I thought it had a Z in it for some reason. Influence. Alright?
That's influenza.
Never influenced by cytokines. Okay? So essentially when the stem cells are made, they're genetically or receptor ball. They have some antigens on them, and we'll talk about that in just a little bit, but they don't have certain receptors. Does that make sense? Because receptors, y'all remember what we talked about with receptors? What are receptors?
Little things that stick out in the back.
That's it. They're these guys, and they're all shaped different based off of whatever chemical needs to bond with them, right? These cells are genetically influenced. And I know, listen, I know y'all are thinking, well, that doesn't make sense. You know what? You're right, it doesn't make sense. Because the way mitosis usually works is this guy and these two should be exactly the same. But they don't. These are the only cells, stem cells in general, but we're talking about HSCs. They're the only stem cells in the body that don't make a true daughter cell. Okay? They make genetically different cells that form receptors around them. Okay?
The HSCs don't have those receptors?
They don't. They're bald at this point.
Where did they stay
What?
at? Like where? Just in the blood?
In your bone marrow. These are all in the bone marrow. HSCs are in your bone marrow. Remember, these are why I said they look like little lymphocytes. Okay? Alright? So therefore, we only need about 20,000 HSCs in our body. Okay? Matter of fact, just about our whole life. Okay? Now, these undifferentiated HSCs, give rise to multi-potential precursors or multi-potential progenitors. Ever how you want to use the P, I don't really care. Okay? These are high self-renewal capacity, which means they go through mitosis a lot. And they can make a bunch of these MPPs. All right? And then these MPPs give rise to all of the lineages of blood cells that we're going to discuss in hematology. Okay? Does that make sense to you guys? All right? Excellent. All right? So, population, about 20,000 sustained throughout the individual's lifespan, as long as everything's normal. Okay? You always got to remember there are abnormalities that can occur. Okay? For instance, like we talked about aplastic anemia. Okay? If something like that happens, something happened and that stem cell lost its ability to make MPPs. Does that make sense? So no MPPs, then you can't make the other lineages. And so your bone marrow essentially dies. Okay? Leukemia, okay? When you have leukemia, you're okay. Even at this point, this guy has a genetic ability to say, okay, I've got enough MPPs for a while. And it stops division. Does that make sense? And doesn't make any more. All right? When people get leukemia or develop leukemia, something happens and it continues to make MPPs over and over and over and over and over and over. Okay? Until eventually you see those increases in white blood cells. And depending on other factors, what it ends up being, what kind of leukemia. Okay? As we go. Alright, so humans contain about 2 times 10 to the 4th, which would be about 20,000. Not morphologically distinguishable, it just means you would have to use something called flow cytometry. So let's stop right there. And I've said this several times, and I know I haven't explained it to you guys yet. All right. So let's talk about flow. It's called flow cytometry. All right. Flow cytometry is a way of identifying a cell, okay? ID cells based off of phenotyping, okay? What is phenotyping?
It's the thing you have to test for you can't see.
So physical characteristics, right? Okay. Phenotyping is physical characteristics. All right. Physical characteristics. Okay. So, on a cell, what makes physical characteristics? Huh?
Morphology.
Yeah, morphology. But what makes morphology? Huh? DNA. How does DNA do it, though? What does DNA do? All right? You know what a dogma is? You ever seen that word? Dogma? Anybody? I think it's Latin. Dogma. I think it's Latin. Dogma means all of the principle, all of the theory, okay, the whole complete thing, okay? Now, I know this may be hard for you guys to understand, but when you finish this, you're going to be a scientist, okay? And you're going to be a good biologist, is essentially what you're going to be. Now, is your degree going to be associate in biology? No. But everything we do is biology, okay? okay? Even the chemistry that we do is chemistry from biology. Does that make sense? Okay? So I'm going to give you some good information. If you guys, have you got biology degrees? I was thinking of somebody who had a biology degree.
She's a math.
She has a math degree.
Okay.
I've had several. I've had biology degrees. I've had chemistry degrees. Those kind of things. All right? The dogma of biology. That's the dogma of biology. The DNA makes RNA, RNA makes proteins. Okay? So when we talk about phenotypes, we talk about physical characteristics. But just about every physical characteristic that we have in our body is caused by what? A what? Well, yeah, it is DNA, but what does DNA do? Why is DNA known as the code of life? Because DNA makes proteins. And without proteins, you die. Did you know that? If you couldn't make proteins, you would die. You know why? Because we have these really, really specialized proteins. They're called enzymes in our body. And if we don't have enzymes, we die. Because just about, I'm going to say every, but just about, I would say probably 99% of the metabolic reactions that take place in your body require an enzyme to make it happen. Okay? So, phenotypes, which are physical characteristics, are caused by what? Proteins. Yes, they are. Okay? So, flow cytometry is taking a cell and we're looking at the proteins that are in that cell. Now, those proteins may be in different forms, but your cell membrane is full of proteins. Okay? So, I'll give you a little advanced before summer. Okay? The summer you guys are going to take a class called immunology. You know what immunology is? Study of proteins. It's actually the study of antigens and antibodies. Okay? But guess what those both are? Proteins. Okay? They're types of proteins. Alright? Antigens are what's on the cells. Okay? All around the cell. Antigens always react with antibodies to form a complex. Okay? So in flow cytometry, we introduce a bunch of antibodies. That are specific to antigens on cells. Okay? So somewhere in a lab, they created antibodies to proteins that we know are on top of that cell. Okay? Now, it gets a little bit more complicated than this, but this is a really, really good review, or a good analogy. Okay? So, when these guys combine, okay, so all antibodies have this shape. They're Y-shaped. They have two heavy chains and two light chains. Is what they're called. Okay. They have a area here and here that can react with proteins. Based off what this area is chemically, is what determines if it's IgG, IgA, IgE, IgM, and IgD. Okay? Those are your 5 classes of antibodies. Okay? Based off of what is here chemically is what determines what they are. Okay? It also determines what antigen it can react to. Okay? So remember those B cells that we talked about yesterday in class that are living in your lymph nodes, and some of them are in your bone marrow. And we talked about the dendritic cells that bring the NARC. It brings a piece of the antigen or a chemical signal of the antigen. Then it stimulates that B cell to become active. Okay? Then it becomes active. During the process, it matures into a plasma cell. Then that plasma cell matures into antibodies. Well, those antibodies have been chemically designed in this area here to respond to whatever that dendrite cell brought to them. Does that make sense? So what they do in a lab is they take B cells, and it sounds crazy, they grow lymphocytes in a culture, and then they expose those to certain antigens on these cells. Okay? So maybe we're going to isolate that protein on that cell. Okay? That makes sense? And we're going to expose those B lymphocytes to it, then it's going to produce antibodies that will attack that particular protein. Okay? And it'll do it they do this for a bunch of different proteins that are on that particular cell. Okay? And the way that they do it, and this is going to sound crazy, but what they do is they basically isolate a bunch of cells from bone marrow. And then they add chemicals that they know will destroy certain cells, and they kill those cells off. Until eventually all they have left is the cell they're trying to isolate. And it's that particular cell. Okay? Then they take that cell and they expose it to those B cells. And those B cells think, oh, hell, that's not my cell. You know, that's not the normal cell that I should have. So it's a foreign particle to it. So then it automatically starts making antibodies to lots of different proteins that are on that particular cell. Then they go in and harvest those antibodies. Then they attach, and this is going to sound crazy, then they attach some kind of chemical to it that either fluoresces, okay, it glows. Once the energy of that antibody attaching to that antigen creates enough energy that it causes that particle to fluoresce and glow. Does that make sense? Okay? So then, you have a machine. Okay? All machine hematology analyzers work kind of on this principle. It's known as the impedance principle. Maybe an E. Impedance. Maybe an E there. Okay? So essentially what happens is you've got a hole. Okay? And you've got a tube that comes through. And then you've got an electrical impulse. Electricity running through this area. Okay? Well, the way that machine works is basically it lines the cells up and it shoots them through that impedance. So what happens, there's an instrument down here that's detecting that electrical impulse. Okay? That electrical energy that's going through. So once a cell goes in here, what's it going to do? It's going to block that electrical impulse. Because look, it's in the middle of it. And so the impulse can't go through. Okay? But then as soon as it goes through here, the impulse starts back. And so this down here basically is saying, stop, 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 stop. So every time it stops, it knew that there was a cell went between it. Does that make sense? It broke, impeded the electrical flow. Does that make sense? So this is how your cell counters work in every hematology analyzer on the market today. Okay? This is the basic theory of cell counts. Okay? So every time a cell goes through, it breaks the pulse of electricity. Does that make sense? So the machine down here is looking for that energy, and it doesn't get it. And so it says 1, 2, 3, 4, right? Every time a cell goes through, and it'll give you your count. Of course, this is going on like instantly, so fast, computer-wise. Okay? Now we've got advanced. We still have this same principle, but now there's like little cameras and little lasers coming in that when that cell impedes the electric flow, it's also looking at for a nucleus, it's looking at its size, and it's looking for granules and the size of those granules inside it as it goes through. Does that make sense? So that's what gives us our diff. Neutro, EO, baso, mono, lengths. Okay? And now you get something that's called immature. Okay? A lot of your instruments now, they actually do a 6-part diff. They'll do 5 parts. Well, actually, they do 7. Because most of your instruments nowadays can actually detect nucleated red blood cells. Okay? And it's all based off of the size and shape of the lasers and how they interpret the results. So like, immature cells are always larger. The nucleus is usually larger also. Those kind of things it takes into account, and it says, hey, that was a metamyocyte, that was a myocyte. And it throws it under this immature. And if you get a certain level of immatures, then it's going to flag the computer to say, hey, that's abnormal. You need to look at that picture. You need to look at a slide on that particular patient. Does that make sense? Okay? Now, that's kind of off subject, but it's not really. So, flow cytometry works the same way. The only difference is those antibodies have attached to proteins, and they all have these little glowing fluorescence on them. Okay? So now what happens is they're going through the little aperture, it's looking for the intensity of the glowing. Does that make sense? Okay? So what's going to happen now, instead of you getting numbers, and you actually will, but you're going to get plots, scatter plots, based off of a graph of size and then number here. Number and then size. And the size we're looking at is the size of the fluorescence. Okay? And so what you'll do is you'll get areas that are cells that are being picked up. And so what we look at is like, say this area here is monoblast. Okay? So we're looking to see a patient comes in and runs CBC, they've got 100,000 white count. We do a slide on them, look at it, and it's all blast. And you can tell they're immature, but you looking at it, it's hard to tell. You can't tell, actually, a monoblast from a myeloblast. Okay? You just can't anymore. So when I was in school, years ago, back in the 90s, and before that, you would have to do a bone marrow aspiration on that patient, and you would have to do these stains. They're called cytochemical stains. And so you would do that, and based off of those stains, you could determine if it was monoblast or myeloblast. Okay? So another way, but we'll talk about that later. But today, we can do flow cytometry. So we can introduce that bone marrow, because that's where those cells originated from. If they're in your peripheral blood, they had to have came from the bone marrow. So you get bone marrow aspiration. Remember we watched a guy do the bone marrow aspiration the other day? And he drew some out, and he put it on slides, and they made slides. And then he came back, and he cored it out, and then he put another syringe on it and pulled a bunch of it out, like 5 or 10 cc's. And he said, this is for flow cytometry. That's what they were doing. They were pulling out that bone marrow, and they were going to send it to a lab that does flow cytometry. Okay? Because they want to know the numbers of the different populations of cells that are there. Okay? Yes?
Do we use flow cytometry in our
No, you will not. And that's what I'm going to tell you guys. you guys want to learn to specialize in something, flow cytometry is what you need to learn to specialize in. Okay? Because that's where we're going. That's the technology that we're going. That's where everything's going to eventually end up being, is in flow cytometry. Okay? Especially with leukemias and stuff. Okay? So what happens is, if our patient has monoblast, this area here is going to be increased, and it's also going to be a brighter color because of those interactions with those fluorescent antibodies on the antigens of the cells. Okay? It's known as phenotyping. Okay? It's also known as like genetic typing because genetics give us proteins also. All right, questions about flow cytometry. All right, got me a break for a minute? What time is it? What? 9:05. Okay. Y'all need just about a 5-minute break, or can y'all? We're just going to go to 9:50. Okay. You want to take a little break if you need to. Pause this. All right, so let's try to finish up a little bit about, at least about the stem cells today. I won't keep you much longer. The stem cells reside in a unique stem cell niches in the bone marrow, so those little microenvironments. They're close to the osteoblastic niches. Remember, osteoblastics are those bones, cells that help us make bone. Everybody get back in here. So they hang out with those guys. So the osteoblast will produce some cytokines that influence the self-renewal of that. So you understand what self-renewal means? What self-renewal means, essentially? It means it has the ability to make itself again, right? So, like, we talked about there's 20,000 of them, about 20,000. So let's say that one of the guys is like me, he's old, okay? And he's not making good MPPs anymore. So let's say that this time he only made one instead of making two because he just got the inefficiency, okay? So what happens, our body is designed to cause something called apoptosis. Apoptosis is essentially cell suicide. It basically tells the cell you're old, you're not doing your job anymore. Then the vacuoles inside it, like lysosomes and peroxisomes, basically burst and it kills itself inside the cell. Okay? And so that cell dies. Okay? Well, as soon as that cell dies, now we have, and that's not exact, I'm just, exaggerating, okay? Now we only have this many, but we want the other one. So what happens is, one guy over here that's new, pretty young, he will go through mitosis, but it's only one cell division to make another HSC. And he says, I'm just going to make a clone of myself. And then it makes a new one. That's what self-renewal is. Okay? Does that make sense? Okay? All right, so stem cells have 4 possible fates. Quiescence, this is that word that I was talking about I'd put on the board in a little bit. Quiescence, it means just to hang out. What is it my kids say? They're vibing. They're just vibing. That's what my 16-year-old says all the time. I'm just going to vibe, Dad. It's like, well, go vibe, dude. And so just hang out. So they're not doing anything at this point. They're just resting. Or they can be going through the self-renewal process. Once they reach a certain age, they go through the self-renewal. Or they can start the process of commitment to differentiation, which means making those MPPs and then taking them all the way to one of the 7 cells that we've talked about. Okay? Or they can go through apoptosis and they're inefficient, not doing their job anymore. That's what happens. Okay? Questions about either one of those? Alright, cool. So progenitor cells. Alright, so upon commitment to differentiation, the HSC enters the progenitor cell compartment. The HPC is a separate compartment. So basically, you've got your compartment here that's got all your HSCs in it. Okay, they're hanging out around those osteoblasts if they're close to the bone, the endosteum. Then, now it's time for us to make some cells. So what happens is, one of these cells moves out and moves to the progenitor compartment. Okay? And then when he does, he goes in here and then starts making MPPs. Okay? And then, of course, those would divide and so forth and so forth. Okay? So they're in their little niche. So you got a little niche that's got a bunch of MPPs in it. Okay? Now, these MPPs can be influenced by cytokines or growth factors, GFs. Okay? They then move out and they go to either the erythroblastic island, the granulocytic niche, lymphoblast, okay, megakaryocyte, whatever, okay, after the MPPs have been influenced, okay. So then they're going to become basically another area that's going to be that CMP, or an area where it's going to be that CLP. And then they're going to go into what they need to do. Okay? Based off of cytokines. Why do you think we get cytokines? What stimulates cytokines? Macrophages, and then other cells too that do this. Because remember we talked about EPO comes from where? Kidney, right? Were you here that day? You may not be here. Was you here that day I talked about erythropoiesis? So EPO comes from your kidneys, all right? So why would those tissues produce cytokines? Because why? You don't know? Anybody know? So why would your body produce cytokines? What would be the basis of making cytokines, certain cytokines?
Red blood cell production.
Why would I need red blood cell production? To breathe. But I'm just normal. I'm just sitting here. Just sitting here on my desk. Right? Why would I all of a sudden need more red cells? From being active? Yeah, that's a good thing. Okay. Let me ask you this. Is being active normal? No. So what influences cytokine production most of the time? Abnormalities. Okay? So things are normal, we just kind of do our thing, we make a little bit, we'll put a little bit of cytokine out there to influence it. But when we start really producing lots of cytokines to produce tons of cells, it's usually due to some abnormality. Okay, so like you said, I need more red cells, I need more oxygen because I'm active. Why would I need more red cells? Well, I flew to Denver today. I got to fly to Denver tomorrow. And tomorrow I'm going to see the town. I'm going to go out and walk up and down the streets of Denver, Colorado. What's going to happen that very first day that I'm there? That's it. Oxygen standard, air standard, right? Because I'm so high up in elevation. And my little old level 200 feet above sea level here in Gadsden, Alabama, ain't getting it done, you know? And so what's going to happen is my body's going to say, my brain, my nervous system, and all the systems in my body are going to say, hey, crap, man, we're not getting enough oxygen. So it's going to say, hey, kidneys, we need you guys to kick out some EPO. And then that EPO is going to get sent to the bone marrow, and then it's going to start influencing those. One of them HSCs is going to go to the progenitor niche, and it's going to start making MPPs. And as soon as it makes MPPs, cytokines are going to go there, the EPO, and it's going to say, hey, look, man, we need some red cells. And so then those MPPs, based off of being exposed to EPO, are going to say, hey, we need to go to that CMP, that common myeloid progenitor, because that's where we make red blood cells at. And then they're going to go to that CMP is going to go to the erythroblastic island, and then get more cytokines from that mother cell, that macrophage, in that particular region. And so then those red cells are going to mature, and that's what's cool about our body. We can speed up the maturation process based off of what we need, okay? It ain't like our kids, you know, our kids are born and we want them to hurry up and grow up, you know, and get out of the house. It takes time. But for our body, we can actually add more and more cytokine that promotes the growth a lot faster. And they can mature a lot quicker. And that way we can kick their butts out by the next day, you know, 2 days at the most. So as I'm there in Denver, the first day may be hard on my body. Second day will be easier. By the third day, if you run a CBC on me, my hematocrits went up high because now I have more red blood cells because my body's keeping them out. Okay, that makes sense to you guys. Okay. And we always have, and that sounds crazy, but if we've got that erythroblastic island, you know, we've got those immature cells right up against the macrophage, there's always some going to be in different levels. Does that make sense? There's always going to be plenty of cells at different levels, so all I really have to do is influence these on the outer level to grow a little faster and mature a little faster, and then I can kick their butts out. Does that make sense? Okay. All right. So, daughter cells, we talked about daughter cells. These are the MPPs. Gradually become restricted in differentiation potential to be one cell line. That's what we were talking about. They leave the progenitor, become a CMP or an CLP, okay, as we go. All right. So the progenitor cells basically make up about 3% of your total nucleated cells in the bone marrow. They do transit cells without self-renewal. In other words, once they reach a certain point in the MPP, they're committed, and they're going to become one cell or the other. They don't self-renew only to a certain point do they go through mitosis. Okay. You can't really tell the difference in them. They look very similar to lymphocytes, just like the other ones. And then growth regulators, glycoproteins or cytokines influence them to differentiate. Okay. All right. So progenitor cells are able to produce colonies of cells. Okay. So a while ago I talked about making antibodies in a lab. Okay. So what we make is we make cultures of cells. Okay. So we may only harvest 1 or 2 or even 3 B cells from a source, so like a blood type or a blood sample. Okay. What we do is then in the lab you can actually introduce colony-forming units, which are chemicals or cytokines that we know will form certain cells. And then you can influence those cells, those B cells, to mature or go through mitosis and make more B cells. Okay? As you go. Now, listen to me closely. Lymphocytes are about the only ones you can do that with. Okay? Neutrophils, basophils, eos, just those 3. Your granulocytes, you can't do that. Okay? So the way you would have to do that is you'd have to harvest bone marrow and be able to get like the myeloblast, myelocytes, okay? Because those three cells are still mitotic. They can still go through mitosis. Once it reaches the metamyelocyte, the band, and the seg, they can't do mitosis anymore. They're terminally differentiated, okay? They can't go any further and can't make any new ones, all right? But other cells we can, all right? So two colony-forming units that you guys need to be familiar with. You do need to know these two, okay? GEMM, okay? Granulocyte, erythroid, myeloid, megakaryocyte, monocyte is what that stands for, okay? I said myeloid, but the G gets the myeloid because it's the granulocytes, okay? Granulocyte, erythrocyte, megakaryocyte, monocyte, okay? So this particular group of cytokines, known as colony-forming units, influences these, okay? Then you have MK, which is megakaryocytes. And right there it was. I don't know why I should have just read ahead. I was doing it from memory. Okay? All right, but that's good for y'all. It is written there for you. Okay? So do know that they are colony-forming units. And these are basically a group of cytokines that can influence the growth of these particular cells. Okay? Maturing cells. Maturing cells constitute about 95% of your total hematopoietic precursor cell pool. Okay? So what are these? These are committed cells in transit population. In other words, they're unipotential. They can only make neutrophils. They can only make eos. They can only make basophils. Okay? That's what I was talking about a while ago when I said that myeloblast, promyelocyte, myelocyte, it can make more cells, but they're going to be neutrophil, basophil, eo. Does that make sense? Same way with the lymphoblast, prolymphocyte, okay? And even some lymphocytes can do mitosis, the mature cell, okay? But all the other cells, not necessarily, okay? All right, so maturing cells are morphologically recognizable, okay, which is very, very important to us. They're measured by nuclear and cytoplasmic characteristics. So what did I do with you guys with our sheet? What did we go by? Cytoplasmic characteristics, didn't we? Okay. Now I mentioned nuclear stuff also in the thing. I just didn't really put it on the sheet. But in class that day we did. We talked about the nucleus. All right. One of the things I mentioned mostly was that NC ratio, nuclear to cytoplasm ratio. Okay. You have an increase in nuclear cytoplasm, so in other words, more nucleus than you have cytoplasm. That's a really good indicator that you're dealing with an immature cell, okay? With the exception of lymphocytes. Lymphocytes tend to have more nucleus than they do cytoplasm, okay? Earliest recognizable cell is known as a blast, okay? Now, listen to me closely. When you get into the hospital and you're working in the hospital, you're not going to have to distinguish if it's lymphoblast, myeloblast, erythroblast, ruboblast, any of those things. In the lab, it's just called a blast. And if you find a blast, then we have to give it to that guy that makes $400,000 a year. Let him distinguish what it is, okay? Now, there are going to be things that you as a tech will know and be able to recognize, and you will know that it's a lymphoblast. You'll know that it's a monoblast, and you'll know that it's a myeloblast, and you'll know it's a ruboblast, okay? Because your teacher is going to make sure you know those characteristics, okay? But when you report the diff out, it's going to be blast, and that's it. That's all that it's going to report to the doctor, okay? Here's why. Because you don't want to have to go to the doctor and say, oh, well, those are lymphoblasts. Oh, those are myeloblasts. Because they're going to say, how do you know? And I'm say, well, dumbass, I learned what they are, But you can't do that, okay? So to prevent confusion, doctors are taught that if a blast shows up, that is abnormal, okay? And that is leads or possible leukemia things that are going on, okay? So that's all you need to be able to do in the lab once you get into a lab, okay? But you also have to be able to recognize that it is a blast. All right, and that's where my job comes into effect. All right. Questions about that? Okay. We actually may have a test Monday. We may be done with this today. Okay. All right. So this is a comparison of your precursor cells as you go. Okay. All right. So stem cells are about 0.5% of your total precursor cells. Progenitors about 3%, and then maturing about 95%. And this kind of gives a little comparison of these. So this will be a good table to kind of look at and make sure you kind of understand what it's talking about as you go. Okay. Precursor cell model. Okay. So precursor cell model is very, very important. So basically what it's talking about is just like what it says. All right. So biology. There was an experiment a long, long time ago, and I don't remember exactly when. It was in, I think, the 1800s, which is kind of scary. But we didn't realize there was a point where we didn't realize cells came from cells. Did y'all know that? We just thought they just freaking formed, you know, as you were growing. So, and I don't remember the scientist's name, but I do remember the experiment. So this guy proved this, that things don't just spontaneously appear, okay? So what he did is he took these jars and he put meat in the jars, okay? And he run a control, right? And his idea was that maggots just can't form spontaneously. They had to come from somewhere, okay? So what he did is he put meat in all the jars, left one open, and then he covered one and then left another one open, okay? And essentially what he proved is that through theory and through hypotheses and then eventually research is that flies had to produce the maggots. Because the meat that was covered never got maggots on it, okay? But meat that was uncovered always got maggots on it, and they couldn't figure it out. They just thought the maggots just spontaneously happened.
Was he in a lab?
Well, probably, yeah, like a little small lab.
Flies get in the lab.
Well, I mean, he was a scientist. He wasn't like an MLT. I mean, he wasn't doing that kind of stuff. He was just a scientist in general. So he introduced the flies, I guess. Or done his experiments outside. I don't know. I'm just telling you. So that was like one of the very first experiments that proved that things just don't spontaneously occur. Okay? So before that, everybody just thought, you know, cells just spontaneously showed up. You know? They didn't realize that there was a precursor cell. There had to be a cell to get another cell. Does that make sense? So that's where this model comes from. And what the theory that we have today is that we call it the precursor. There has to be a precursor of everything. Okay? You know, I don't know if you guys believe evolution or creation. I ain't going to get into that with you guys. But evolution says that, you know, the reason we have a mitochondria is because billions of years ago, a little single-cell organism got a bacteria called it. And then when it replicated that bacteria. So eventually it said, hey, I'll just keep it, and kept it going. Same way with chlorophyll. And their justification of that is if you guys don't know, you actually have two genomes in your body. Y'all know what a genome is? Have you ever heard that term? What's a genome? You know what a genome is? A genome is all your genetic makeup. Okay, so in us, normally we think about our nucleus. All the cells that have a nucleus in our body make up our genome. But we actually have two genomes. Every cell has nuclear genes, but there are also mitochondrial genes that we have. And those mitochondrial genes actually come from our mother. So when you get down to DNA testing and parentage testing, that kind of stuff, a lot of times what they're actually looking at is mitochondrial DNA because it's more specific. Because you guys, I don't know if you guys know this or not, you guys, every one of you in here, ain't none of y'all kin, are you? And I asked that because Abby and Aaron are aunts and niece. Did y'all know that in the other class? Y'all know Aaron, don't you? The older lady, blonde-headed, sits in the back usually. And Abby, that's their aunt and niece.
Is that second?
They're not, huh?
Is that second?
Yeah, the ones that are in clinicals now.
Oh,
Okay? But you guys, I don't know if you know this or not, but you're all 99.9% the same. Did you know that? Yeah, you are. Every one of them. I take your DNA, and every bit is. So what that means is, that's what makes us different in our DNA. 0.1% of our DNA is different. Okay? But mitochondrial DNA is easier to compare and look at the differences in, because you get that from mom. And so, it's very easy to see the difference there. All right? So earliest differentiating daughter cells are the CLP and the CMP. I've made mention of those several times today. Those are basically the ones you need to know. So common lymphoid progenitor, common myeloid progenitor, and this tells you what these give rise to, these particular cells. And we've made mention of this already also. But there it is in writing so you guys can see it. Okay. So what do cytokines do? Cytokines are also known as growth factors. They're basically, you have colony stimulating factors, which are what those form those colony forming units that we talked about a little while ago. That's their job. Okay. The cytokines that we're real concerned with in the lab is interleukins. Okay. Interleukins. And these interleukins, they play a role in white cell production, but they also can play a role in red cell because they interact with this CMP. Okay? Those interleukins and those other cytokines, this is where they begin to interact, right here, in the CMP level and the CLP level. Okay? Because based off of our body's need, we're going to give certain cytokines or interleukins to make these things go. Okay? There is a chapter or a page in your book. Is this not your hematology? I got one. No worries. I got one. Got it out right over here too. On page page 46 of your book, table 4 or 5 is a big list of different interleukins or different cytokines that you guys kind of need to at least look at, okay? Need to kind of be familiar. Most interleukins are IL with a number, okay? And a lot of interleukins interact with lots of different cells. Does that make sense? So in other words, they're multi-influencers, okay? Multi-potential influencers. So some of them only work on lymphocytes, some of them work on lymphs, monos, basos, that kind of thing. And the page that I told you guys, 46, has what they do on them. It's a big list. Do you have to memorize that list? No. But at least kind of look over it and have an idea so you'll know what we're talking about. Okay? All right, produced by many cells, but macrophages are one of the most common cells that we see. Endothelial cells, which are the cells that line the walls of vessels and also the bone. Osteoblasts and those adipocytes, or those fat cells. We talked about that earlier. Okay. Most are produced by the stromal cells in the bone marrow. Those little microenvironments, with the exception of erythropoietin. There's erythropoietin. I knew I don't spell it right the other day. But there it is in black and white, also known as EPO. And this is produced mainly in the kidney. Okay? I think your liver can produce a little bit of EPO, but it doesn't produce enough to maintain your body like it needs to. So that's the reason you see when people go into kidney failure, you see a decrease in EPO. Okay? Most are not lineage specific. This is what I was talking about. They're actually multiple function, potential chemicals, so they can affect lots of different things. Okay. So how do cytokines work? Cytokines work by receptors on the cells. Okay. So this is the reason those HSCs are not really affected, is because they don't have true receptors on them. Okay. But then when they get to the MPP and the CMP and CLP, then they begin to develop receptors. And so then they can be influenced by cytokines as they go. Okay. A lot of these cells, their growth requires it for continuous growth. This is a lot of things that are being used in treatments for like leukemias and stuff. Okay, so if you have leukemia and you have a particular cell line, and we know what cytokines are necessary for that cell to grow, they can give you drugs that block those cytokines from being produced, and so therefore that cell won't continue, and it'll eventually die. And so this is a big research thing that's going on right now with chemo drugs. Okay, as you go. All right. Negative regulators. Y'all guys know what positive and negative feedback systems are, don't you? Okay. So that's kind of what this is talking about here. A lot of the cytokines can act as negative regulators. They can stop the growth or they can increase the growth depending on what it is. If I've got a cell that doesn't need to replicate and it keeps replicating, is that a negative regulator? Yes, it is, because it's doing something that's abnormal. So when we talk about negative regulators, we're talking about things that can cause abnormal stuff to happen, okay? All right. Talked about that microenvironment, and that's it. So we will have a test Monday, okay? So let everybody know. Of course, we'll come in and we'll take our test, and then we'll start the next chapter, okay? Any questions about anything? Yes, ma'am. No. No, Monday we're going to have it because we got to move on. You know, we've got a lot to cover, and this next unit is really big, and it's got a lot of information in it. So I mean, I'm willing to wait till Wednesday, but we're going to have class Monday, and we're going to start new material. Y'all want to do that? That's confusing to some people. I mean, I'm just saying. All right, how many want to take the test Monday? Okay, that's the majority of the class, guys. All right, so test Monday. You've got your study guide, baby. It's not like it's going to be hard. I mean, y'all take it in 15 minutes, you know, like you've been doing. Yes?